In this video, we will discuss about one of the pancreatic cancer treatment idea that can be used, and if this idea works on the uh, mouse model, then there is a good chance that this idea might also work on the humans. So, before going into this idea, let us quickly revise what are the main points that we get from the literature about pancreatic cancer. So, the recent studies of the pancreatic cancer says that in pancreatic cancer there is a great perturbation in the glycomics profile, especially the O-glycans that are formed in the pancreatic tumors uh, very widely compared to those of the healthy pancreatic cells. So, uh, from this point, one hypothesis can be made that there is a change in the O-glycan genes that are responsible for glycosylation in the pancreatic tumor cells. So, th- this hypothesis will be used in this for generating this idea, and if this idea is successful, then we will have more insight about which genes get disrupted in the pancreatic cancer, or some of the genes that get disrupted in the pancreatic cancer, and that might leads to a novel treatment strategy. So let us not waste our time and go through this strategy in brief. So uh, the one should or the study begins with the pancreatic cells. So in this case, two pancreatic cells are being considered, that is FG colo 357 and CD18 cells, and then uh, viral transduction of these cells or individual cells will be done with different libraries. One library contains the essential genes that are responsible genes that are essential for the genes that are essential for the survival of the cells, and simultaneously there is another library that is transduced in another set of pancreatic cells, that is O-glycan library, and this O-glycan library contains the genes that are responsible for O-glycosylation. So in total, the four library will be generated for this FG Colo 357. The two libraries, one library that contain essential genes, and one library that contains O-glycan genes. And same things are done with the CD18 cells. After that, after the library has been prepared, then cells are being sorted based on the lectin bindings, and as lectin binding differs into the cells, as most of the cells or most or some of the cells that does not contain this that does not contain some of the O-glycan genes will produce a different O-glycosylation profile, and that will be reflected in the lectin binding. So based on the lectins that are responsible for O-glycosylation and other glycosylation, this cell sorting will be carried out, and then cell will this amplification of the cells will be done in the cell culture medium, and after the sufficient population of cells are being produced, then this cell population will be injected into the mice to see which mice will produce the effects that are similar to the pancreatic cancer, or is there any pancreatic cancer symptoms formed in those mice or not. And suppose one, as there is a high probability that one of some of these mice might form or might show the pancreatic cancer, as the previous literature shows there is a perturbation in the O-glycosylation, and we have used one of the library as the O-glycan library. So after this one of the after the mouse shows the pancreatic uh, tumors, the there are two kind of things need to be taken care of at that point of time. First one is that the immune cells or the immune T cells need to be isolated from those from that those or from that mice that shows pancreatic cancer, and simultaneously their cells need to be analyzed to see uh, what kind of change in the glycomics profile occur compared to the healthy mice, and from that analysis we can take one of the unique glycan that is formed in the mice that is affected with the pancreatic tumor, and that is not present or present in very minuscule amount in the mice that are healthy. So that unique glycan can be used as a antigen for the CAR T cell plasmid, and this plasmid then be translated into the immune T cells to form a CAR T cells, that is chimeric antigen receptor cells, and this chimeric antigen receptor cells will then be transduced into the pancreatic mouse to see whether uh, there is a reverse action of these pancreatic cells occurs within the mouse or not. And if the pancreatic cancer has been reversed in those mouse, then uh, this model could be if also effective to treat the pancreatic cancer in the humans. And in order to use these CAR T cells, there are different kind of CAR T cells that are being evolved, and some of them have shown positive results even on the solid tumors, not just on the uh, homeopathic, not just on the blood cancer cells. So in this, one of the important thing that we need to take care of is that use the CAR T cells. Those are effective on the solid tumors, as this pancreatic cancer is a solid tumor cancer, and there we need to take care of two major things. First one is the tumor microenvironment, another is the accessibility of the antigen against which this CAR T cell is developed. So, and there's some T cells have shown positive results and has been able to overcome this kind of problems. So we can use those CAR T cells in this study and to check whether this idea works to treat the pancreatic cancer in the mouse model or not. So I hope you got a brief idea how this idea works to treat the pancreatic cancer. And if you if you need to understand more about this pancreatic cancer treatment idea, you can go through our previous videos where we have discussed about how these solid tumors, what are the difficulties that occurred in treatment of solid tumor compared to the like other tumors such as blood cancer tumors. So that's all for this video. Thank you for your time.